Hello, beautiful people. Today we're looking at homeostasis, which is actually one of my favorites. There's not many slides, but don't let that fool you. This topic is not easy, especially for those in HL. Let's dive in. So, the dog is here to warm you up to the topic. This is easy. Homeostasis is just maintaining the internal environment of an organism. Easy, okay? Like, it's keeping variables such as temperature and blood pH as optimal as possible. Good. Now, we're going to look at several examples. The first one is temperature. There's a lot here, so let's go through it slowly. Okay, so this uses negative feedback. You need to understand, by the way, just in general, that negative feedback is used in homeostasis as a key mechanism. Why? Because when a variable, like body temperature, strays from the set point, the optimal point, negative feedback always changes it back. So it brings it back to baseline, right? On the other hand, positive feedback pushes conditions further away from the norm because positive feedback amplifies in loops, right? Whereas negative feedback, again, brings it back to a base point. In humans, our most stable internal temperature is around 37 degrees, and this is controlled very, very efficiently. So how does it work? Well, first of all, we have central and peripheral thermoreceptors. So peripheral thermoreceptors are found in the skin and they respond to external temperatures. And central thermoreceptors are found, for example, in the hypothalamus. And they monitor the core body temperature, right? Whereas peripheral monitors external temperature. Okay, so when the hypothalamus detects that the body temperature is too high or too low, it's going to activate things to correct it. And we're going to see. First, let's say it's too cold, so body temperature suddenly falls, and that's detected. The hypothalamus is going to coordinate vasoconstriction, which is reducing blood flow to the skin. That minimizes heat loss. Shivering, because that generates heat through involuntary muscle contractions. Increased metabolism, which raises internal heat. Here is how increased metabolism works, by the way. The hypothalamus, you need to know all of this, unfortunately. The hypothalamus releases TRH, which stimulates the pituitary to release TSH. That stimulates the thyroid to secrete thyroxine, which boosts cellular respiration. And the cellular respiration means more metabolism, means more heat. I'm sorry you have to remember all of that, but you do. And then also another thing that can happen is uncoupled respiration in brown adipose tissue, which produces heat. Again, also hair erection, right? So goosebumps. Okay, what else? When the body is too hot. So when the body temperature rises, we basically do the opposite, right? So vasodilation, meaning more blood flow to the skin to promote heat loss. Sweating to cool the body. In this case, water evaporates from the skin surface, right? And so the hypothalamus can stimulate our glands to release up to 2 liters per hour. And reduced metabolism. Okay? And this basically will result in us going back to the normal body temperature. Okay? So it's energetically costly, but it allows a stable internal environment for cells. Hope this is clear. Next thing, blood glucose. This is very easy. So again, we normally maintain blood glucose at around 5 millimoles per cubic decimeter. Don't worry about that. And we use 2 hormones, insulin and glucagon, okay? Both are secreted by the islets of Langerhans in the pancreas. So first, let's look at the situation when blood glucose is high, seen here, okay? When blood glucose is high, what happens is insulin is released. And insulin makes cells take up glucose from the blood. So, for example, the liver, right? And they convert it into glucagon, meaning in the blood, the glucose decreases back to normal. What about when the blood glucose levels decrease? Then we produce glucagon. Oh my God, sorry, before I meant to say glycogen, okay? Liver cells turn glucose into glycogen. Okay. When the blood glucose is low, we release glucagon, which is the enzyme. And glucagon helps the liver release glucose. Alrighty? So, 2 separate processes, but the purpose is the same, to return it to base levels. Also, I'm just realizing this slide is wrong, by the way. In this case, the liver does not break up glycogen to release glucose. It's the opposite. The liver makes glycogen from glucose that it's taking from the blood. So many issues here. Okay, And then diabetes is related to these two things. So diabetes is a failure to control blood glucose, basically. And type 1 is autoimmune, meaning our own body destroys our beta cells. Remember, beta cells are the ones that release insulin, so no insulin is produced. And this appears early in life. Type 2 diabetes is insulin resistance, meaning we actually do produce insulin, but the target cells, these here, right, don't respond properly. And that basically means it's pointless, right? Because we're producing insulin, but then the blood glucose levels are not decreasing. Okie dokie. That's it for you SL people. Unfortunately, the ones in HL, you have to stick around a little longer. So let's get started. First, osmoregulation. This is a beast of a subtopic. It took me so long to understand this in IB. So don't worry if you find it tricky. All right. You have to understand that the kidney does homeostasis in two ways. It does excretion and osmoregulation, right? So it removes toxic waste, but it also controls the osmotic concentration of body fluids, okay? So let's look at the steps one by one. Here is the summary of the whole thing works, right? And then I've segmented into parts. So part 1 is ultrafiltration and reabsorption. What happens here? So this is where urine actually begins to start formed. So we're in the nephron, by the way. And ultrafiltration is when the glomerulus, okay, forces plasma through fenestrated capillaries into the Bowman's capsule, which is this thing in yellow. And that forms the filtrate. So a lot of things pass through to the Bowman's capsule, okay? Except proteins and cells, which stay in the blood, but most other things pass through. And then in the proximal convoluted tubule found here, selective reabsorption occurs. So sodium goes back by active transport, glucose and amino acids go back by co-transport with sodium, and water follows by osmosis. So 80% of water and solutes are reabsorbed here. Okay, so a lot passes through here, but then a lot of it goes back. Then, once it's passed through here, whatever it hasn't been reabsorbed, meaning it's going down, goes down into the loop of Henle, which is this. This is the part that confuses most people. Simply put, okay, the loop of Henle creates a constant, like a gradient, okay, a gradient of sodium for later. We'll talk later about why that's important. But in the descending limb, okay, this is permeable to water, but not salts. So water exits, okay? And then in the ascending limb, later on, this pumps sodium out, okay? But no water leaves. So what you actually need to know is that you get this very steep gradient, this osmotic gradient, okay? So then, and this is where it's important, in the third part, okay, in the collection duct, it's called, water is reabsorbed. But the gradient that we formed before allows for water to be reabsorbed. All right. So how is water reabsorbed? It depends on ADH. So ADH is this hormone, which depending on how much water you need, causes more or less water reabsorption. So basically, if there's high blood osmolarity, right? So it's very concentrated. That triggers osmoreceptors in the hypothalamus, and that signals to the pituitary to release ADH, okay? ADH, what it does basically is it inserts aquaporins into the collecting duct. So here, right, on the collecting duct, if you have ADH, you're going to have aquaporins here, so more water is reabsorbed. But remember, this water is reabsorbed via osmosis, and that's why this gradient is important, okay? If blood osmolarity is low, meaning there's not a lot of concentration in it, ADH secretion falls, so less aquaporins and more dilute urine. I don't know if this explanation makes any sense or if you're more confused than before. Any questions or dilemmas or, yeah, if you're crying over this, just leave it in the comments. I'll reply and help you out as much as I can. There we go. The last thing is blood supply to organs. This is really easy, okay? Obviously, the amount of blood your heart can beat is limited. So the body has to redistribute blood between organs depending on activity. I think that's quite easy to understand. And this is done by vasoconstriction and vasodilation of arterioles, right? So a narrowing an arteriole reduces blood flow, widening it increases it. So I'm going to give you an example, and then the rest you can just read because this is not difficult at all. So during vigorous exercise, you're going to have increased blood flow for muscles, makes sense. Also to the brain to support alertness, but less to the gut and kidneys. Why? Because even though these are necessary processes, when you're sprinting, maybe they can wait a little, basically. And the same goes for sleep and wakeful rest. We have different priorities at any given point. Let's do questions, my favorite part. So, which of the following best describes negative feedback in homeostasis? I think this is pretty straightforward. 3, 2, and 1. Pause now. So, it reverses a deviation to restore balance, right? It takes it back to baseline, okay? A response that amplifies a change from the set point is positive feedback. And then a constant output regardless of input is no feedback. And B is just BS. Okay. The next is, which change would you expect during dehydration? And again, 3, 2, and 1. You would get more insertion of aquaporin into collecting duct membranes. Why? Because you'll have increased ADH. Why? Because you're dehydrated, so you have high osmolarity in your blood, meaning you have a lot of solutes in it. Straightforward enough. And lastly, which of the following changes would decrease heat loss from the body on a cold day? And once again, 3, 2, and 1. You would constrict arterioles in the skin, right? On a cold day, you don't want to lose heat. You're cold already. So you don't want much blood flowing to the skin because that causes heat loss. So you're going to constrict the arterioles so that there's less blood flow. Again, I think simple enough. Okay, again, any comments, send them my way. I'll see you next week for the next topic. Bye, everyone.